human genomes, mutation hotspots identified scientists have discovered that specific regions in the human genome are unusually prone to mutations, particularly at the points where DNA is actively read and copied. These so-called mutation hotspots occur at transcription start sites, the locations where RNA polymerase opens DNA to initiate gene expression. The finding offers critical new insight into how genetic diseases develop and evolve. Why this matters understanding these hotspots is essential because mutations are the driving force behind both genetic disorders and adaptation. While most mutations are harmless, a significant number contribute to rare genetic conditions affecting an estimated 300 million people globally. Accurate models of mutation rates are vital for studying these disorders effectively. The new research highlights a previously overlooked mechanism: the inherent vulnerability of DNA during the process of transcription. The vulnerability of transcription, the process of copying DNA into RNA, exposes the genome to increased damage. Imagine a cookbook with recipes, genes. When the book is opened to copy a recipe, transcription, the pages are susceptible to tears or stains. This analogy explains how repeated opening and closing of DNA during transcription can lead to imperfect repairs, resulting in permanent genetic changes. The researchers estimated these events occur hundreds of thousands of times per cell daily. Conflicting data and mosaic mutations, initial analysis of long-term inherited mutations, extremely rare variants or ERVs, revealed a clear hotspot at transcription start sites. However, when the team examined de novo mutations, DNMs, those that appear randomly in offspring, the hotspot vanished. This inconsistency puzzled researchers until they considered mosaic mutations, errors that occur during early embryonic development. Mosaic mutations are often overlooked in standard DNM studies because they appear patchy and can be mistaken for sequencing errors. Once the researchers re-examined data including mosaic mutations, the hotspot reappeared, confirming its existence. This discovery revealed a blind spot in current mutation research methodologies. The mechanism behind the hotspots, the researchers concluded that the constant opening and closing of DNA at transcription start sites creates a fragile environment. The RNA polymerase, while essential for gene expression, can misfire or leave DNA exposed too long, leading to damage that isn't always repaired cleanly. This is a missing piece of the puzzle about where DNA mutations come from, and one that may improve studies of genetic conditions relying on de novo mutation data. The study suggests that future research should account for mosaic mutations and re-examine discarded data near transcription start sites to improve accuracy. In conclusion, identifying these mutation hotspots provides a crucial step toward understanding the origins of genetic diseases and refining the tools used to study them. The findings emphasize that DNA is not a static code, but a dynamic, vulnerable system subject to constant wear and tear, especially at the very sites where life's instructions are read.